last class on Tuesday we have finished with the conversation about the synapse. And I just want to remind you a few things about the structure. When we talk about synapse, it's a point of interaction between two cells, correct? There are two major types that I mentioned to you. The electrical synapse, which enables communication between the cells by the means of ionic flow. And ions are exchanged from one cell to another and back. And this synapse can work in both directions. Chemical synapse, the one that we're going to talk about the most, is generally found around the around the nervous system. It is the only type of the synapse that neurons are communicating through. And in chemical synapse, the impulse, the signal, goes only one way from presynaptic to the postsynaptic neuron. And signal transmission is mediated by the release of the chemical, so-called neurotransmitter. So let's talk about the steps of the synaptic transmission. So few terms that we are using here may sound unfamiliar, and that's perfectly fine, because we're going to talk about membrane physiology. Hopefully we will start to talk about it today, just a little later. So I choose to start talking about synapse first, figure out how the signal goes through, and then talk about the details of that transmission. First of all, I want you to appreciate that the membrane of each cell has a certain electric potential. Okay? We're going to talk about how this potential is formed, but so far, understand that if you take a cellular membrane, that lipid bilayer, okay, then the outer side, sorry, I'm going to erase it and just make it more visible. So if you have a lipid bilayer, then the outer side will be positive, the inner side will be negative. And that difference in charge will make the potential, it is the cause of potential. That's what potential is. When you have two different opposite charges on two sides of the membrane. Okay? So when electrical impulse travels through the axon, and electrical impulse on this picture is shown with this purple arrows. This electrical impulse, as it travels through the axon, is called action potential. Okay? Pretty much it's an electric current that represents the nerve signal. It's the electric current that can be transmitted to the next neuron. Is that clear? That's the signal. That's what we call nerve impulse. That's the thought. That's the command. Okay? So when action potential arrives at the presynaptic terminal, that structure of the axon, okay? So this is that structure, the presynaptic terminal. When action potential arrives there, the distribution of electrical charges across the membrane changes. Do you agree with me? So we have certain electrical impulse on the membrane, like on the wire. Does that make sense? The change in the potential on the membrane is called depolarization. Membrane becomes more positive. We'll come back to that later. Okay, so membrane becomes depolarized. There's a change. And that depolarization is a signal for calcium channels to open. Now, if you don't mind, I want to give you a quick reminder about the channels. Okay, let me see if I can do a little picture for you. So that would be, I think it would be worthwhile to speak about the transport across the membrane. Okay. I'm going to open the notebook and make some drawings for you. So before we start talking about the transport across the membrane, what is the plasma membrane consist of? What does it consist of? Can anyone tell me? Those are not sperm cells, by the way. Those are molecules that plasma membrane consists of. Anyone tell me what they are? Anyone? Okay. They have hydrophobic and hydrophilic parts. And what are they? Anyone remembers? Phospholipids. Very good. So which part is hydrophobic? The outside. So this part of phospholipid. So did I say hydrophobic? Yeah. Hydrophobic is inside. Sure. Yeah, that's hydrophobic. I'm sorry. What about the outside? Hydrophilic. Yes. So, which molecules can travel across the membrane without any problems directly? Like this. Should they be hydrophilic or hydrophobic to go through the membrane directly? Think about it. You have a membrane, okay? It's hydrophilic on the surface, but hydrophobic on the inside. Can you tell me why? Not only this, what does it do? What does the membrane do, except for the transport? Yes, it's a barrier between what and what? Excellent. Between the cytoplasm and internal and external environment, right? So it's a barrier between what's outside of the cell and what's inside. What is the major component of the cytoplasm? Yes, it's water. What's the major component of extracellular fluid? It's water. So you have to separate water from water. If you just put hydrophilic barrier, it would get dissolved. So I got to put something that would repel water. And that's going to be your hydrophobic component, hydrophobic inside. But that hydrophobic inside will not be able to stably exist in the watery environment. So you cover hydrophobic barrier with a hydrophilic surface. Does that make sense? Now, considering this, that you have hydrophobic inside, it's like, you know, like you go to the party where you don't like anybody. You don't act like a total asshole. I mean, you can think of all those people whatever you want, but you usually wear, you know, this nice mask. You know, you communicate with people. You can hate them. It's not a problem. Usually, do not express it if you want to be socially acceptable. So same here, you know, inside it's hydrophobic, but on the outside it's hydrophilic. So which molecules can cross the membrane? Hydrophobic ones, because hydrophilic will run into that wall, right? Hydrophilic would run into that wall. So hydrophobic, pretty much meaning lipid-soluble molecules. Molecules like respiratory gases, oxygen, CO2, various small organic compounds, for instance alcohols, can easily cross the membrane. If there is a need to transport compounds that are hydrophilic, like ions, potassium, calcium, charged compounds, they cannot cross the membrane, so they have to get across it using different types of transmembrane proteins. Is that clear? So these transmembrane proteins can be channels. Does that make sense? Channels can be leaky, and we're going to talk about it, or they can be gated, which means they are normally closed, but in response to a certain stimulus, they will become open. Does that make sense? Okay, let's go back to our synapse. So when action potential arrives at the presynaptic terminal, change in the membrane potential works as a signal for calcium channels to open. And calcium starts to diffuse into the neuron. Why does calcium diffuse into the neuron? This is a simple question. It's not a tricky. Why does calcium diffuse from outside of the neuron to the inside? Say again? It is, but why doesn't it go backwards? Why doesn't it leave the neuron? Keyword here is diffusion. So calcium is high where? Outside and low inside. That's why it flows in. Does that make sense? It's the only reason. So when calcium gets inside of the cell, it stimulates the intracellular pathway. I'm going to tell you something. Calcium is the important intracellular signal. Okay? I mean, it works as the signaling ion very often. So what happens? Influx of calcium into the cell stimulates the transport and fusion of the vesicles, so-called synaptic vesicles, with the membrane of presynaptic terminal. And when these vesicles fuse, they release the content into the synaptic cleft. Does that make sense? So calcium here works as a trigger. Are we clear on that? We good? Okay. So these vesicles transport to the membrane of presynaptic terminal, fuse with the membrane, release the content. Their content, those tiny green dots here on this picture, content is called neurotransmitter. It's the chemical. So when neurotransmitter is in the cleft, it diffuses across the cleft and interacts with its receptors on the postsynaptic neuron, sometimes called postsynaptic terminal. Does that make sense? Binding to those postsynaptic receptors, neurotransmitter binding, causes certain changes. For instance, neurotransmitter here can open channels on the postsynaptic membrane. or it can initiate so-called second messenger system activation. Does that make sense? So it's all good. Neurotransmitter interacts with the receptors, causes certain changes in the postsynaptic neuron. What do you need to do with neurotransmitter in the end of the process? What do you think? When you're calling someone, what does your call end up with? You hang up. So in this case, because otherwise another person would be, talked to death, right? It's impossible to speak forever. So neurotransmitter, its action should be also terminated. Otherwise, it will overstimulate postsynaptic neuron. Does that make sense to you? Effects of neurotransmitter can be terminated in 3 ways. It can diffuse away from the synapse. As you can imagine, it takes a while. Does that make sense? It can be taken back into the presynaptic neuron, or can be destroyed. Does that make sense? Couple more things about the whole process. First of all, the amount of neurotransmitter that is released is proportional to the amount of calcium that enters the cell. Imagine this. If the signal to open calcium channels lasts for a very short time, then the amount of calcium that enters presynaptic terminal is small. The amount of neurotransmitter that is released is small too, right? So changes in the postsynaptic neuron will be relatively small as well. If action potential, if the change on this neuron, if the change lasts for a long time, that calcium channels are open for a longer time, more calcium flows in, more neurotransmitter is released. Does that make sense? More neurotransmitter, stronger, more significant, profound changes in the postsynaptic terminal. So essentially, the amount of calcium regulates the strength of the signal. What is neurotransmitter? Where does it come from? What do you think? Simple question. It is what? What does presynaptic neuron? What does it do? Where does it come from? The neurotransmitter. Really? What is the cars on the streets? Where do they come from? They just miraculously appear out of the thin air? They're made. Neurotransmitter is made, right? Neurons make neurotransmitter. And that's the point, because if the signal is too strong, okay, if there's too much stimulation, then neurotransmitter storage can be exhausted. Pretty much presynaptic terminal runs out of neurotransmitter. Does that make sense to you? This is called synaptic fatigue. And that's actually good, because it provides certain level of negative feedback regulation. If the signal is too strong, to avoid overstimulation of the neurons downstream, next neuron, postsynaptic neuron, if neurotransmitter is gone, there's going to be no signal transmission and, no overstimulation. Does that make sense? Here's what I want to do with you. you can use your recordings, you know, slides and whatnot. I want you to kind of walk with me through this process, and I'm going to do like sort of a flow chart. I'm going to give you a hint of what you can do at home as a study tool. Does that make sense? So what does it start with? The transmission, synaptic transmission. The signal, we call it what? Action potential. Where does it arrive? Which one? Is it before synapse or after synapse? Where does it arrive? Before, yes. So it's at pre-synaptic terminal. Okay? This action potential, what does it do? What does it open? Not the sodium. Opens calcium channels. Okay? What does it lead to? Opening of calcium channels. Say again? Yes, so influx of calcium. We're going to use influx, the word influx, in the reference of the ionic flow into the cell, and the word efflux out of the cell, just to make it easier, okay? Just pretty much for the rest of the course. So we're going to talk about ionic flow a lot. Now, what does calcium do? What is its role? So, release of neurotransmitter. I'm kind of skipping on the moment when vesicles go to the membrane and fuse with it, right? But please keep it in mind. They don't just release neurotransmitter. They have to get to the membrane first. Does that make sense? Now, where is neurotransmitter? Where is it getting released? Into what? Bless you. Does that make sense? Okay, and what does neurotransmitter do next? It interacts with receptors on or at of which neuron? Postsynaptic. Does that make sense? And what is that? What is the effect of this interaction? Okay. Inhibition or excitation of postsynaptic neuron. We good on that? Now, neurotransmitter, at this moment in our story, neurotransmitter, where is that? Where does it hang out now? Some of it, yes. Some of it is in the cleft, right? So what do we need to do with neurotransmitter? Get rid of it. How? diffusion, degradation, or reuptake. Okay? Does that make sense? Bless you. The neurotransmitter, obviously, is the molecule that's responsible for the transmission of the signal. If you can somehow increase the amount of neurotransmitter in the synaptic cleft artificially, what's going to happen to the signal? If you increase the amount of neurotransmitter in the synaptic cleft, what's going to happen to the signal? It's going to be stronger, yes. Don't hesitate to tell me. Okay, now look at this scheme over here and tell me, how can you increase the amount of neurotransmitter? Go ahead. Okay, yes, you can increase amount of calcium. In terms of therapeutic intervention, that would be not best approach, because you have to increase it systemically, and that may have really lousy effects on many other organs. Okay, but calcium, yes, you can do that. How else can you increase the amount of neurotransmitter? Any wild guesses? Can you some other way increase its release? Well, that's an interesting way, but probably so. There are drugs or toxins that increase the release of neurotransmitter. You can just, independently of calcium. Does that make sense? Can you increase this? Sure. What else? For calcium? Yeah, that's pretty much increase the influx of calcium in any way. Increase calcium or increase the influx. Yes. Those things are the two that we mentioned so far, calcium influx and calcium independent release of neurotransmitter. Those guys are hard to manipulate. There are a few things that are easier to mess with. How can you increase the amount of neurotransmitter in the synaptic cleft? That can be pretty specific. I pretty much hear your brains like rotating and working. Calcium, it's also release. Try get away from releasing calcium. Excellent. You can prevent neurotransmitter from being destroyed. If you can, diffusion you cannot really inhibit, but diffusion contributes very little, okay? But if you can get rid of degradation or reuptake, you can increase the effects of neurotransmitter. And when we will talk about neurotransmitters, I'll give you examples of the drugs that mess up with that. Okay? That make sense? Now, this whole kind of flow chart, I strongly recommend you do that, because I will test you on every aspect of this pathway of synaptic transmission. It's going to be a big deal in the exam, okay? I always tell students, when I tell you you must know that, it means you wake up at 2 a.m. in the morning and you recite the whole thing. Not by memory, you can like draw it, okay? That's the level of knowledge I want you to have. I don't care that much about brutal memorization, But this, it's a must. Because what happens is, a lot of the knowledge about synaptic transmission, depolarization, repolarization, action potential, ionic flow, then is carried over into the AP2. And if you don't have really profound knowledge of this here, it's going to be a struggle there. Really, it's basics. Okay, did I scare you enough? I hope so. As you can imagine, the entire process requires a lot of ATP. To give you a perception of how much ATP is required, your brain is about 2% of your body weight. It consumes 15% of the energy that you take in with the food every day. You know, so it's remarkably important. Now, receptors that neurotransmitter interacts with. There are two types of receptors. One is ionotropic. So you can see them here, ionotropic receptors. Ionotropic receptors open ion channels. Here on this illustration, you can see the red neurotransmitter that binds to the receptor. Receptor is essentially part of the ion channel. Does that make sense? Okay, we good? When receptor binds to the ion channel, it works like a key. It opens the ion channel, and ions, shown in purple, they diffuse into the cell. Or out of the cell, for that matter. It depends on which ion or which channel is opened. Does that make sense? So, if neurotransmitter is excitatory, it opens sodium channels. When sodium gets into the cell, sodium is positively charged. So what happens to the charge of the cell? It becomes more positive too. So this is called excitation. Okay? When neurotransmitter is inhibitory, then sodium channels are not open, but potassium or chloride channels are open. So what happens, either chloride ion gets into the cell, chloride is negatively charged. What happens to the cell charge? More negative. So membrane potential of the cell becomes more negative. This is called hyperpolarization. Cell becomes inhibited. Another option is to open potassium channels. Potassium concentration inside of the cell is higher than outside. So potassium will start to diffuse out. If cell starts to lose positively charged potassium ions, what's going to happen to the cell charge? More negative again. Cell becomes hyperpolarized. You get immediate inhibition. Does that make sense? Now, important thing about ionotropic receptors is that they are very, very fast. Okay, so that excitation or inhibition can happen in milliseconds. Does that make sense? So these guys, that's neuronal transmission, quick transmission between the neurons, fast. Now, metabotropic receptors work in a different fashion. They are often called G protein coupled receptors, or GPCR. What does that mean? When neurotransmitter, again shown in red, binds to the receptor, on the cytoplasmic side of the receptor, which is transmembrane protein, there's an assembly, the protein that's called G protein, and it gets activated. So when G protein gets activated, it initiates the whole chain of signaling. Okay? that we call a second messenger. Okay? Second messenger is produced at some sort of a molecule, and that second messenger molecule then may eventually interact with a channel, open the channel, and cell gets say depolarized. So I'm going to illustrate this idea by doing a little trick. Kelsey, can you do me a favor? Can you dim lights a little bit? I think it's can you turn them off? Just turn them off. Just turn them off. That'll work. Yeah, just turn them off. Okay, no. It's probably something with the lights. Then can you do something else for me? Open the door. Thank you. So in this case, go ahead, open. So Kelsey and the door, and can you just leave it open? If you just press it, it's going to stay open. Yeah. So thank you, no problem. So Kelsey worked as, so I was an ionotropic receptor. And Kelsey worked as the ion channel. It was quick, right? I told her to open the door and she opened it. Now channel is open. Does that make sense? This is the second messenger. Deliver it to Kelsey, please. I don't really care the pathway. Okay. Don't read it, just get it to her. You can read it. Okay? Now, was it quick for me to tell Kelsey, thank you very much, to tell her to open the door? It was quick. How long did it take to tell you to close it? Quite a while, right? So, second messenger system takes longer time to respond because of all those multiple steps. And everybody who was in the chain of getting this message to Kelsey was a part of that second messenger system. At this point, I do not require you to know the steps of second messenger system. We will get into it as we talk about each separate one. But all I need you to understand is that you have very specific G protein, which is cytoplasmic protein, which initiates the whole signaling. Does that make sense? So in this case, for instance, if I'm a receptor, then Ashley is G protein. She started it. You see what I'm saying? Understand the difference, okay? Between ionotropic and metabotropic receptors. Are we clear on that? Now, I want you to understand the effects of both of them can be the same. They both can lead to excitation or inhibition. It's the time of the response. All right? Now let's talk about neurotransmitters. And it's going to be a lot of fun because we're going to talk about some drugs. It looks like a lot. Don't be too afraid of it. I'm going to prepare a little sort of a quiz, kind of, that you can use as a practice tool. Okay. So there are two major types of neurotransmitters. Small molecule and neuropeptides. So small molecule neurotransmitters, they are rapidly acting, but the response that they induce, they last for a short period of time. The response is acute. Does that make sense? And the reason for it, if you think about it, when the small molecule gets in the synaptic cleft, it can diffuse out of the synaptic cleft much faster than when it's a large molecule. Does that make sense? So let's talk about small molecule neurotransmitters. These guys are more common in the nervous system, okay? And some of them you know pretty well. So ACH, acetylcholine. Quite frequently I'm going to use this acronym, it's just easier, okay? So acetylcholine can be excitatory or inhibitory. I pretty much highlighted. I don't really care if you know or you don't know about which receptors it works as excitatory or inhibitory with, but just know that it can work as both, okay? It's one of the major neurotransmitters in the central nervous system, in the cortical area. So it's one of the major neurotransmitters for your thoughts. And it's also a critical neurotransmitter in parasympathetic nervous system. Sort of a glimpse, parasympathetic nervous system is the system of relaxation, so-called rest and digest. Okay, when you sit down, you know, you digest food, your pupils constrict, you read the book, something like that. Okay. Now, what's interesting about ACH is that you can modulate these effects. Remember, we were talking about the inhibition of, if you inhibit degradation of neurotransmitter, you will increase its effect. Have you heard about organophosphate pesticides? Well, I didn't expect anyone to heard about organophosphate pesticides because it's pretty specific for horticulture. Organophosphate pesticides are pretty commonly used in agricultural practices all over the world, and they in fact were used since practically the beginning of 20th century. Unfortunately, in 1930s, German scientists, and I have to give kudos to those scientists, you know, they were smart. They noticed one thing, that farmers who used organophosphates, they frequently came down with the very interesting poisoning symptoms. So those German scientists thought, can we make it stronger? And in the form of gas. And they managed to do that. Have you heard about the gas like sarin? I can write it down for you. Sarin or VX. VX is the most toxic chemical weapon that is known to humanity. Potentially the most toxic chemical that was ever invented by humans. It's a neurotropic gas which causes symptoms generally called sludge. Sludge stands for salivation, lacrimation, urination, defecation, gastrointestinal cramps, and emesis. Emesis is vomiting. Now, all those things, you know, separately aren't really pleasant if they happen in excess. Now imagine all those things happening to a person all at the same time. Kind of lousy, right? So what happens? Those gases, sarin and VX, they inhibit the enzyme called acetylcholine esterase. That enzyme, really long name, that enzyme destroys acetylcholine. If you block it, acetylcholine doesn't get destroyed and keeps stimulating glands and muscles, okay? And the death is pretty grim, and it happens pretty quickly. Does that make sense? the mechanism. You inhibit degradation, overstimulation of downstream neurons, the person dies. Alright? Are we cool? Any questions? Another interesting toxin that messes up the ACH effects is the toxin coming from the microbe called Clostridium botulinum. It's a botulism toxin that inhibits the release of acetylcholine from the neurons, and it causes paralysis because acetylcholine is the only neurotransmitter that stimulates the contraction of the muscles. As far as I remember, it happened about 3 months ago, this summer. A family in California, they bought few salads from the gas station. Mother died, father and kids ended up in intensive care and barely survived. They ingested botulism toxin. When that type of paralysis, so-called flaccid paralysis, happens, when muscles cannot be contracted, there's one critical muscle in your body. If it stops working, you die. Voluntary muscle. It's not heart. Heart is not voluntary. That's voluntary muscle. Yeah, your diaphragm. You die of respiratory arrest. That make sense? Now, another interesting drug is curare, which South American natives used to put on the arrows. It was really nice, you know, even if it's a small wound. Curare gets in the bloodstream, inhibits the receptors, completely paralyzes the person dies. Another drug like this is atropine. And that's the one that I wanted to point your attention at a little bit more. Okay, so if you don't mind, I'll go back to the notebook and we're going to discuss the mechanisms by which neurotransmitter interacts with the receptor. Well, not so much mechanism, but how you can mess up those mechanisms. So this is like a generic receptor, okay? And this is neurotransmitter. Does that make sense? And they are interacting. So far, no questions, right? So, what can you do to block the interaction? What should you inhibit? Hard. Extremely hard. Small molecule, it's hard to block it. You can block it. Does that make sense? So you take, say, an inhibitor, okay, and block the receptor. Does that make sense? Okay? Is that clear? Now, I have another question for you. Receptor, neurotransmitter. Is there any way you can increase the signaling? What if you will provide a molecule that will not be a neurotransmitter, but will still bind to the receptor? Can you do that, hypothetically? So this inhibitor will block signaling, and the stimulant, sorry, that was the mistake, and the stimulant will increase signaling. Does that make sense? So, terminologically, inhibitor of neurotransmitter receptor is called antagonist. That's in general. Anything that decreases the effect of the receptor, decreases the signaling through the receptor, is called antagonist. And this molecule is called agonist. Does that make sense, this idea? Do you understand what I'm saying? Agonist versus antagonist. Now let's go back to our table. So curare and atropine that block the receptor, what are they? Antagonists. Nicotine that is not acetylcholine but binds to the receptor and stimulates it is what? Agonist. Do you understand that? Now, we're going to have an attempt. I'm going to give you a clinical case. You have a call. Imagine that you're doctors, okay? And you just received an emergency call that in, I don't know, in the main campus, there's a terrorist attack. And people were exposed to the sarin gas. Okay? They're exposed to the sarin gas that causes this through this. Which drug would you administer to the victims of the poisoning? It's right there. Think about what's going on with the sarin gas. You block the degradation. So the amount of acetylcholine in the synaptic cleft is skyrocketing. Does that make sense? So the signaling through the acetylcholine receptor is overwhelming. It's overstimulation of the receptor. What do you want to do? Block what? Yes. With what? Curare would be a little bit tough, but atropine, yes. And actually, in like a medical kit for military servicemen, they have atropine in case of that type of attack. They have to administer it. It's the antidote for sarin. Sarin or VX or any type of that gas poisoning. Does that make sense? Atropine has another interesting effect. Sometimes if you go to optometrist and they need to very thoroughly examine the ability of the muscles in your eye to accommodate the shape of your lens, they need to relax those muscles completely. And it's really hard to do. Because you use those muscles, because when you change the focus of your eye, you use those muscles constantly. So what sometimes they do, they give you some drops, pretty much they put them right there, and those drops lead to complete blockage of acetylcholine receptors in the muscles that change the shape of your lens. Okay? The side effect is that they also completely relax muscles that constrict your pupil, so your pupil become dilated for about 2 or 3 weeks. The drug atropine can be isolated in nature from one plant. I don't remember the Latin name for it. And in 18th and 19th centuries, the extract of that plant was used by women as sort of a cosmetic feature, should I say. What women did, they used that extract, they put some drops in the eye, and their pupils became dilated. And that was considered beautiful at the time. Okay? The downside, except that you look like a junkie, is if you OD on that, you're going to die because of paralysis. But other than that, you know, it's pretty neat. I actually went through that procedure myself when I was like 10. For 3 weeks, you're not allowed to read. Good luck with that. You couldn't hold me from... Oh no, for me it was 3 weeks. For 3 weeks I looked like I was on heroin every day. Okay. Now, another couple of other interesting effects that are associated with ACH. In Alzheimer's, production of acetylcholine is decreased. We don't know if it has clinical effects. However, it has one interesting implication. It is demonstrated. Now, a little disclaimer, I do not advertise smoking. It was demonstrated that nicotine consumption decreases the risk of Alzheimer's development. Daily consumption of nicotine in the amount equivalent to 3 cigarettes is sufficient to decrease the risk of Alzheimer's by 25%. Okay? First, it does not justify smoking because the risk to develop lung cancer is much higher. Second, you don't have to smoke. You can do like nicotine patch or nicotine gum. Third, you can reduce the risk for developing Alzheimer's simply by doing mental work. You don't have to do a scientist. Doing crossword puzzles will suffice. Any type of mental effort will decrease the risk of Alzheimer's development. It's kind of interesting. Nicotine really decreases the risk of Alzheimer's development. Also, if you ever heard about the condition called myasthenia gravis, no? No? Pretty much when receptors for ACH are destroyed, okay, acetylcholine cannot stimulate skeletal muscle, and it initiates sort of progressive paralysis. It usually starts with inability to elevate the eyelids, condition called ptosis, and then it can lead to further paralysis of the face. Usually starts in the face. Okay, it's autoimmune disease, yes.
So what if like an older person already has Alzheimer's, would smoking like decrease or maybe like set it into like a regression, would do anything, or is it just like a preventative?
If the person has Alzheimer's, then probably any sort of nicotine consumption, including smoking, will decrease the symptoms a little. Does that make sense? Hmm? Mm-hmm. Okay. Go ahead. Keep going. You mean about the mental work? Here's the thing. That's the moment that I took. I think it was last class or class before when I was talking about the risk. When you see patient to patient, you see the cohort that already has Alzheimer's. So for you see 100% of patients that you see, well, not, but a lot of patients that you see have Alzheimer's. Why? Because you work in the healthcare. Healthy people do not come to you. Does that make sense? So if you would take a percentage and you would look at the onset of Alzheimer's in people who do mental work versus people who do not, it's really an established fact that if you do any type of intellectual effort, again, you don't have to be a lawyer. Reading, memorizing verses, doing crossword puzzles, doing Sudoku, anything will delay the Alzheimer's onset. Does that make sense? But it inevitably gets skewed. Does that make sense what I'm trying to say? So like you see people that come in and doctors have Alzheimer's. Of course some of them do. Okay, like you can work at the cardiac rehab facility and say, look, all sportsmen, marathon runners, weightlifters that come to our rehab facility, they all have heart problems. That's why they are in the rehab facility, because they had heart problems. Doesn't mean that doing sport dramatically increases your risk of developing them. Does that make sense? We have to be careful about our perception. You see what I'm trying to say? Now, another very, very important, those are probably two most commonly mentioned neurotransmitters. Another one is norepinephrine. It used to be called adrenaline. Now it's called norepinephrine. Again, can be excitatory, can be inhibitory, depends on the type of receptor. Found again in CNS, also found in sympathetic nervous system. Now when I say inhibitory, excitatory, think about it. EpiPen, everybody knows what's this, right? Now when a person has anaphylactic shock, what happens to the person's blood pressure? It's down, okay? It's down because smooth muscles of the blood vessels are relaxed. At the same exact moment, what happens to the person's respiratory passages? They're constricted because smooth muscles of bronchi are excited and contract. Does that make sense? So when somebody applies EpiPen, which is essentially norepinephrine, smooth muscles of the blood vessels are excited and smooth muscles of the bronchi are relaxed, inhibited. You see? All difference is different receptors. On the smooth muscles, different adrenergic receptors on the smooth muscles of the blood vessels versus smooth muscles of the bronchi. Does that make sense? It's also called feel-good neurotransmitter. Amphetamines increase the release, and drugs like antidepressants and illegal antidepressants like cocaine, they prevent the uptake. So norepinephrine stays in a synaptic cleft for a longer time, therefore its effects are enhanced. Okay, so you feel better. Does that make sense? On the opposite side, there's a drug called reserpine, which treats hypertension. Okay, it decreases the levels of norepinephrine because norepinephrine has stimulatory effects on the heart and blood vessels. When you decrease norepinephrine levels, heart rate goes down and blood vessels become dilated, so it's kind of an acute intervention against the hypertension. But since norepinephrine levels go down, the regular consumption, regular taking of that drug, decreased levels of norepinephrine can lead to clinical depression. Okay? Dopamine, again, either excitatory or inhibitory, found in the CNS, especially in the substantia nigra in the brainstem and in the basal nuclei. In addition to regulating mood, it's another example of feel-good neurotransmitter. It also regulates motor activity. So deficit of dopamine in Parkinson's disease leads to the motor deficiencies associated with Parkinson's. Does that make sense? Since it's feel-good neurotransmitter, everything that messes up its uptake will make you feel better and will make you to fight depression. Okay? Now, this is cocaine, so probably not the best idea to use it to treat depression. Drug like L-Dopa increases the release of dopamine. Have you heard about this drug? L-Dopa is used to treat Parkinson's. And, don't remember, Oliver? No, I don't remember. There's one guy, I need to go back and I will check the name. I will Google the book, okay? He's an American neurologist who wrote the book Awakenings. It's the story of patients that had some kind of viral infection, and years after they recovered from that infection, they had catatonic conditions similar to Parkinson's. You know what catatony is? Person is pretty much irresponsive to the outside stimuli, cannot do anything, you know, sort of a stupor. So that guy, that neurologist, he was the first person to try L-DOPA to treat that type of condition, and he successfully treated many patients. So they awakened from that catatonic condition. Okay. Serotonin is inhibitory neurotransmitter. It can be found in central nervous system and regulates a lot of subconscious activities, involuntary activities like sleep, your circadian rhythms pretty much, appetites, and regulates the mood. So Prozac, the anti-depression drug, inhibits reuptake, okay, increasing the amount of serotonin and treating the depression, as well as MDMA, which works as the receptor agonist. LSD is receptor antagonist, so it may lead to depression if OD'd. This is one of the drugs that you can buy over the counter in the section of what they call biologically active compounds. That snake oil part of any pharmacy, where they sell things, and on the bottom of the bottle you can see the effects are not evaluated by the Food and Drug Administration, which pretty much means this thing is not going to kill you. Other than that, we can't guarantee anything. So if you want to, you know, spend your money on something that has no proven clinical efficacy, go ahead. Now this time serotonin can be bought, and if you take it before you go to bed, it will make you sleep better. Really. Okay, you can do that. The only problem is that if you need to wake up really early, you feel like somebody, you know, hit you in the head. Does that make sense? Histamine can be either excitatory or inhibitory, found in CNS, but not only in the CNS. It is also found in the digestive system, works there as the hormone, regulates digestive activities, but in the brain it regulates mostly the sleep-wake cycle. What is the advice that physicians give to young parents sometimes? And young parents are about to fly with kids like 2-3 years of age. You know? Give them what before the flight? Benadryl. Benadryl is antihistamine, which can be easily absorbed into the brain, and it will block histamine effects, and it will make kids sleepy. Modern antihistamines, for which the main function is what? What do they do? What do they counteract? Antihistamines. What do you take them for? Allergies. Okay, modern anti-allergies, things like Claritin and Zyrtec and whatnot, they chemically cannot be uptaken into the brain, so they do not reach hypothalamus and they do not mess up your sleep-wake cycle. Does that make sense? Gamma-aminobutyric acid, GABA, one of the major inhibitory neurotransmitters in the brain. Certain chemicals enhance its effect. Things like alcohol or anti-anxiety drugs like Valium or barbiturates, they enhance the effects of GABA, and if inhibition starts to dominate between neurons, then the control, voluntary control over the muscles decreases, so motor coordination decreases. Does that make sense? The whole idea, why you become so clumsy when you're drunk. Okay. On the opposite side, if you use the inhibitors of gamma-aminobutyric acid receptors, you mess up with the inhibition of nerve impulses. And when there is no inhibition, overstimulation of the skeletal muscles can cause convulsions. Does that make sense? Okay. Glutamate is the excitatory neurotransmitter. Important in the learning and memory, but interesting feature of glutamate is that it kills neurons in stroke. When there is an ischemic stroke, the lack of blood flow to a certain area of the brain, neurons that are deprived of oxygen and are in the state of ischemia or hypoxia, whatever you want to call it, start to produce a lot of glutamate biochemically, and glutamate overstimulate of neurons, which leads to their death. Okay. Glycine is major inhibitory neurotransmitter in the spinal cord. Have you heard about the drug chemical called strychnine? What is it used for? Generally. No. It's, I mean, I can imagine using it for the heart attack, but that would be sort of a terminal decision. At end of 19th, beginning of 20th century, you could buy it like then analogs of Home Depot. It was a rat poison. Also helped a lot of families, a lot of couples to resolve their problems, either by getting rid of a husband or getting rid of a wife. It's a great poison, okay? It doesn't, as far as I know, it doesn't have very strong smell or taste. So people used to add it to other people's meals. And strychnine inhibited the receptor for glycine, which caused convulsions and permanent contraction of diaphragm, which led to respiratory arrest and death. Right? Now a few more interesting small neurotransmitters. ATP can be either inhibitory or excitatory, depending on the receptor involved in the pain sensation. And you can imagine you cannot really inhibit ATP. If you inhibit ATP, you're boned. Adenosine is an inhibitory neurotransmitter that controls sleep-wake cycle. Chemicals like caffeine, which can be found in a coffee, theobromine, which can be found in the chocolate, or theophylline, which can be found in tea, block adenosine receptors in the brain, and that's why coffee or tea or chocolate have those stimulatory effects. Nitric oxide, well, kind of have to mention it because if you read any physiology literature, it's nitric oxide this, nitric oxide that. I'm not saying it's unimportant, quite an opposite. Nitric oxide can be found everywhere. But the point is, it's not so much neurotransmitter as physiologically active molecule. It's a gas, okay? Produced in the stroke and can kill neurons in the stroke. Can cause vasodilation in the peripheral circulation, so a lot of it. Okay. Now, final sort of a treat for you: endocannabinoids. Those are neurotransmitters that are produced in the central nervous system, and they are inhibitory. Okay. They control memory and appetite. So the drug tetrahydrocannabinol, or THC, that can be found in marijuana, is the agonist of endocannabinoid receptors. Knowing that, you can easily see how some effects of marijuana consumption can be explained by the fact that THC from marijuana stimulates the receptors normally stimulated by endocannabinoids. For instance, increased appetite or inability to properly process thoughts. Does that make sense? We're going to take a break now, and when we will come back, we will talk about large molecules. Before we go on the break, things that you have to know about neurotransmitters are all listed in the study guide. What I told you now is more exhaustive than the study guide. Okay? If you can answer questions in a study guide, you're going to be fine. Another thing that I want to advertise now, before the exam, we're going to do a quick review of the study guide in the class that will be recorded. Does that make sense? To make you feel a little bit better. That doesn't mean that you have to postpone your work on the study guide, which I probably will publish like in 2 parts, till the very last day. Okay? I'll publish the first part today. How about that? Sounds good? Awesome.